Okay, I think we'll go ahead and get started. Good afternoon, good morning, and good evening to you, wherever you might be chiming in from around the world. I am Rob Burgess, and I head up business development for Sinobiological. And I would like to welcome each and every one of you to the next installment in our BioTalk Tuesdays series of webinars, where we hear about cutting edge technology as it applies to basic biology and biological research, as well as translational research for the development of vaccines and therapeutics and even diagnostic kits. We have a fantastic speaker here today who's going to tell us about cancer treatment and how it is being revolutionized with T-cell based therapies. Before I introduce the speaker, I would like to just introduce one housekeeping issue, and that is that I ask everybody, number one, to please type into the chat box hello and tell us where you're from. We're always very excited to see where everybody is calling in from around the world. And then number two, also type your questions in for the speaker into the chat box. We will withhold all questions until the end of the speaker's webinar, and then I will walk through those questions verbally for the speaker and try to get to as many as possible. So in that respect, it is my pleasure and honor to introduce this week's speaker. It is Dr. Crystal Paulos. Crystal is the David H. Lawson MD Professor for Cancer Research at Emory University. She also holds a variety of other titles, including Professor in the Department of Surgery, Microbiology, and Immunology, and Director of Translational Research in Cutaneous Malignancies, and finally, co-leader of the Cancer Immunology Program at Emory. Crystal is focused on translational research and developing new T-cell based therapies for solid tumors. Her lab pioneers strategies to reprogram the immune system to fight cancer more effectively. And her work has advanced into multiple new clinical concepts and clinical trials. She is also deeply committed to mentoring the next generation of cancer researchers and building collaborations that move discoveries from the lab to the clinic. Crystal obtained her doctorate in biochemistry from Purdue University, and she has completed 2 postdocs, one at the NIH and one at the University of Pennsylvania. And the title of Crystal's talk today is Revolutionizing Cancer Treatment with T-cell Therapy. So Crystal, it's an honor to have you today, and I will turn the virtual podium over to you.
Thank you, Rob, for that very kind introduction. Today, I will be presenting to you new work that we've just published in the journal called Cancer Cell, and we're really excited about the work. We have basically, in a Cliff Notes version of this, we've identified a CD4 T-cell subset called a Th17 cell. When it expresses an antigen receptor that recognizes cancer, surprisingly elicits an anti-tumor response directly, but then it induces a host B-cell response. And this is very unexpected work, which has lots of implications for building next-generation cell therapies. This is my laboratory in the middle here. We went axe throwing just as a group to have a lot of fun together. And one of the folks I'd like to feature that was the driver of this project is Anna Cole right here. And I will call out her work throughout this presentation. And we're very lucky to work with the investigators that surround this photo, with basic scientists, translational scientists, surgeons, dermatologists, all of which we work together so we can translate findings at the bench to our patients. My laboratory specializes in a type of a therapy called adoptive T-cell transfer therapy. And the reason that we focus on it is because it can induce remarkable anti-tumor responses in patients. This is a cartoon of what we do. We work very closely with surgeons at Emory that are experts in melanoma, as well as other types of malignancies, such as cholangiocarcinoma, for example. We can get tumor from these patients. We can chop those tumors up into small pieces. And the reason we do that is because there are actually immune cells that live within that tumor. And we can expand those T cells using food called IL-2 cytokine, which causes these T cells within the tumor to expand rapidly, logarithmically. to a level that's quite large. 50 million cells can be expanded from one small tissue that was in a 24-well plate, for example. And then we can cryopreserve these cells and use them in patients, for example, that recur after PD-1 therapy. And this therapy can be really effective. I like to show this particular figure because it's features a patient that had triple negative breast cancer. And before the patient got the adoptively transferred T cells, you can see in these yellow arrows tumors throughout this patient's body. I'd like you to also appreciate within the liver that there's lung metastasis too. However, 22 months after this patient was given this type of adoptive immunotherapy, it's quite remarkable. All the tumor has gone and you can't detect it anymore. And this particular patient has had a long-lived anti-tumor response and, dare I say, was cured. So we also see the same thing, types of potent anti-tumor responses in patients with CLL, given a different type of T cell therapy that probably many of you have heard of, called a chimeric antigen receptor, where we can take T cells and genetically engineer them to be antigen specific and then infuse them into patients. And this has been remarkable. And it has really just changed the way in which patients with hematological malignancies are treated. The point of this particular figure is to show that many of these patients have progression-free survival for years. But unfortunately, you can see that some of these people actually progress. And the goal of our laboratory and many laboratories that focus on adoptive T cell transfer therapy is to find ways that we can improve this T cell product so that we can increase the amount of survivalship in our patients. Our particular passion and focus is on a T cell that's called a CD4 cell, and more specifically, a helper T cell. Most investigators, I would say, that work on adoptive T cell transfer therapy focus on the CD8 T cell, which is shown here in this light blue-purple color. These are effector T cells that can directly recognize tumor or get educated by antigen-presenting cells to be proliferative. We turned our attention to yet another subset, these CD4 cells. And the reason we are so excited about them is because they're called helper cells for a reason. They help the entire immunological community. So we see that they enhance CD8 T cells. They can bolster NK cells that play a big role in eliciting anti-tumor responses. And collectively, they are this kind of the social glue that holds all this immune response together. And so we hypothesized, instead of using CD8 T cells, wouldn't it be cool if we could identify a CD4 subset that might be a major player in eliciting anti-tumor responses? And so to address this question, we turned to a CD4 model where all the CD4 cells have a T cell receptor that recognizes tyrosinase. And the reason we wanted a TCR against tyrosinase is because it's overexpressed on many human melanomas. And so this model is very clinically relevant because it recapitulates that. And this is a melanoma model. So we take these anti-tumor CD4 T cells, which are in a naive state, and we program them down different pathways. Classically to a Th1 pathway or an unprogrammed pathway called a Th0 cell, which have been shown to elicit remarkable responses when combined with CD8 T cells. We also compared a more newly described T cell subset called a Th9 cell that produces lots of IL-9 versus a Th17 cell, which has been very controversial. Some folks have shown that these Th17 cells can cause tumors to grow more effectively, and some have shown that they elicit anti-tumor responses. But to our knowledge, no one has ever compared these different T cell subsets head to head, even though we can generate them. And so Hannah Knockelman, who used to be in my laboratory, who's now at Stanford doing medical oncology, addressed this question. So she programmed them to different fates, and she found that she could do that. For example, these Th1 cells produce the most interferon gamma, whereas the Th9 cells were able to produce the most IL-9. And interestingly, the Th17 cells could produce a lot of IL-17. Convinced that we made these cells, we then asked the question, which of these subsets might kill the tumor better? And quite honestly, when we were doing this experiment for the first time, we suspected it would be the Th1 cells because Th1 cells produce interferon gamma that then can cause the tumor to express more of its antigen via MHC and cause the T cell to regress. Well, this is a cool moment, in my opinion, because we were wrong. And when you're wrong, it's kind of fascinating to really understand why your hypothesis might not have turned out as you expected. Interestingly, we found that these CD4 cells that were programmed down this Th17 phenotype were really good at killing tumors. So let me just walk you through this. The NT means no treatment, and this is a mouse with melanoma. And you can see the tumor grows unabated. The open circle are the Th1 cells, not doing much better. The Th1 cells are highly ineffective, and the Th9 cells only suppress tumor growth slightly. But you can appreciate very visually here by the red triangle that the red diamonds, the Th17s are really robust and elicit long-term responses after these T cells are adoptively transferred. So in digging deeper into this finding, we found that that's not just the case in that piece of data I just showed you, but remarkably that these anti-tumor Th17 cells, if they're genetically engineered to recognize cancer or have a TCR that recognizes cancer, that they are very good at eliciting anti-tumor responses. So I'm just listing 4 different types of malignancies where we see both in mouse models and human NSG mouse model systems that this is the case. These Th17 cells are really good at killing tumor, which begs the question, what makes these cells tick? Why are they so good? What makes them effective? Is it that idea that these T cells cooperate with CD8 T cells? Or perhaps more than that, they cooperate with a community of different types of immune cells? Or in contrast, is it possible that these anti-tumor Th17 cells can do it alone? Because it has been now shown that tumors can express MHC class 2, which is what CD4 is recognized by TCR. And so maybe they can just really kill directly. So Anna was really intrigued by this data, and she asked some really important questions that we've gotten every time I give this talk. These Th17 cells are really effective, and so what if you don't need them to be antigen specific? See, the CD4 cells that we use either have a chimeric antigen receptor that recognizes tumor antigen, or in this case, a T cell receptor that recognizes tyrosinase overexpressed on melanoma. But what if these T cells didn't express that, but they were Th17 cells? Would just globally any type of Th17 cell do this? Maybe it doesn't need to be antigen specific. And then she also wanted to know if maybe all of these Th17 cells, maybe it's all about the host B cells. And so it could be a generic Th17. So we found that, yes, the T cell has to be tumor specific. So if we program Th17 cells that don't recognize tumor, which is this Th17 cells from C57 black 6 mice that don't have antigen specificity, the therapy doesn't work. Or if we program OT2 Th17 cells and put them in an animal that doesn't have ovalbumin expressing tumor, the therapy doesn't work. Only in the cases when the T cells can recognize the tumor by way of MHC. So we think this is kind of how it's playing out, that of course Th17 cells, even in the absence of B cells, are important. They proliferate, they can directly kill tumor, but it's not enough. We must engage the host B cells. And when we do that, these B cells can proliferate, they empower the Th17 cells, but not only that, they start producing these tumor-specific antibodies. And these antibodies decorate the tumor and cause some form of tumor destruction, maybe ADCC. So is that true? If we take the peripheral blood, the antibodies from the peripheral blood, would they bind that tumor? What Anna did is she took animals that were long-term cured animals from this Th17 therapy and compared them to black 6 mice. By the way, this Th17 therapy also causes robust autoimmune vitiligo because hair tissue also expresses, hair follicles also express TRP1. Anyway, I'm sorry. We took the peripheral blood from these animals and we assumed that they would have antibodies. And if so, they would bind the B16F melanoma tumor. And then we could use a sandwich assay and flow cytometry to see if that's the case. And what she found, you can see with this IgG below, that the antibodies bound B16F10 very well if they came from this cured animal, but not at all if they came from an animal that didn't get this therapy. And so we saw this across the board. It was specific. These antibodies detected the melanoma that these animals were given, this B16F10 melanoma that grows really aggressively. And to a lesser extent, much less, these antibodies really didn't recognize other malignancies, including melanoma. There was this very low level of binding to B2905, which is a melanoma, HC Mel 1274, which is a melanoma, and Yummer 17. We also saw that didn't bind to pancreatic cancer or to healthy immune cells. There was something about, these antibodies had to recognize the tumor in which they destroyed. So visually, this is what we think is happening. These Th17 cells that we can, by the way, produce and within 4 days, so you can imagine the clinical relevance of that. We make these cells and 4 days later, our patients could get them. When we infuse them into the body, they engage with B cells. That empowers them, but then the B cells also empower them to proliferate. And then it secretes these antibodies. And so if these antibodies are actually important, we thought, wouldn't it be cool if we could take the antibodies themselves and put them into another mouse, and maybe they would protect against, for example, lung metastasis. And that's exactly what Anna did. So this experiment is definitely a labor of love. You can see the following. We give a mouse melanoma. We then precondition with total body radiation or lymphodepletion, which is used in the clinic, and that's to empower the adoptively transferred Th17 cells to expand, proliferate. And in many of these animals, they're cured. So 100 days later, Anna bled these mice, took the serum from these mice, and then put them in a brand new mouse. So this serum was given to animals that are C57 black, 6 mice, and then we give a tumor. And we ask a question about protective capabilities. Does the serum, which has these antibodies in itself, reduce lung metastasis? And that's exactly what we found. So if you give a lung metastasis to an animal, within about 21 days, you can see lots of this lung met burden throughout the entire lung. If we give serum from an animal that was age matched but didn't get this Th17 therapy, there's still a lot of lung mets. In contrast, you can see in this red, which I'm going to go to first, the serum from those animals really reduced the amount of lung metastasis the animals got. And TA99 is just a control. It's a known antibody that binds tyrosinase that has been already clearly shown to reduce lung metastasis. So there's something in the serum. We're not sure what those the antibodies are recognizing, but that bind B16 tumor and reduce lung metastasis. So this is really our whole story that I really want you to take home. Adoptively transferred Th17 cells, which are in red, interact with host B cells. These host B cells in part act as antigen presenting cells to drive these T cells to proliferate even more by presenting antigen. They also interact with class 2 and CD40 signaling that cause more activation. And then these Th17 cells produce IL-21, et cetera. And these B cells then as a consequence proliferate more. They detect antigen themselves, get educated, and some of them class switch and produce tumor-specific antibodies that bind the tumor and kill it. And so it's a 1-2 punch, in our opinion, that both the cells themselves as well as the host B cells are important for this anti-tumor response. So how does our work impact patients is really where we're going next with this work. And so we hypothesize that maybe in situations where patients recur, for example, patients with melanoma that recur from checkpoint blockade therapy might be great candidates for this type of therapy. And that we could treat them with this potent T cell product. And perhaps not only would the cells elicit a direct anti-tumor response, but they would inspire host B cells and humoral immunity to kick in. And so as a consequence of that, a very talented scientist in my lab, Megan Wyatt, is really driving this. She works very closely with the surgeons at Emory, and we've looked at the ability to make these types of T cells from patients with oral cancer, melanoma, prostate cancer, cholangiocarcinoma, etc. And what we've seen universally, which is surprising to us, that compared to high dose IL-2 expansion strategies, which is the conventional strategy, using this programming of this type 17 phenotype, we're calling them polar TIL, expand even better. And we see this across the board. Each of these dots is a different patient tumor. And we get equal amounts of CD4s and CD8s. So we're really excited to turn this product into medicine. That's not the only way we're doing it, by the way. We're also interested in sorting Th17 cells based on chemokine receptors as well as co-stimulatory molecules. We have published a paper showing that T cells that have this Th17 phenotype in a CAR model elicit better responses against pancreatic cancer too. So we're turning this into a clinical trial in collaboration with our friend Carl June at the University of Pennsylvania, and our collaborator, Ned Waller, along with myself and Greg Lazinski. So I will just end with this. We really think that this interaction is important. There's something special about helper cells that produce IL-17 and probably more importantly, IL-21 that causes this interesting downstream effect. And that B cells, which we really didn't appreciate before, are extremely important for this outcome. I'm happy to take any questions. I want to first thank Anna Cole, who did all of this elegant work, along with our collaborator, Hannah Knockelman, who was a former student in my laboratory, and all of our funding sources, and a really close collaborator of mine, Greg Lazinski. And I'm happy to take any questions, and thank you for your attention.
Thank you, Dr. Paulos. That was a fantastic presentation. We sure appreciate that. Very eye-opening. We already have some questions from the audience, so I'll just jump right into those.
Should I stop sharing or just keep it here?
No, keep it up in case you have to go back to a slide.
Okay, sure.
A lot of times
Will
that
do.
happens.
All right, thanks.
So Ranjan Ramasamy is asking, do the activated B cells act as better antigen presenting cells against the Th cells? Do you understand that?
Yeah, I do. That's a great question. So they do. We've sorted B cells from an animal that got the Th17 therapy compared to B cells from the animal that didn't get the Th17 therapy, and then, sorted them and then compared them head to head in their ability to act as antigen presenting cells by way of causing the T cells to proliferate. And we found that the B cells really did proliferate better if they came from the animal that got the Th17 therapy. And also, which is shown in the paper, these B cells have much higher levels of class 1, class 2, MHC class 2, CD80, 86, CD40, etc.
Great, thank you for that. Omar Kessler is asking whether the CDR determination within the microenvironment and introduce the antigen could contribute to improving immunotherapy responses. CDR determination within the microenvironment and then the introduction of the antigens could contribute to improving immunotherapy responses.
I think I understand this question. And if I'm totally off base, please, Omar, please email me. But that's a very good question. I think you're asking, does the type of tumor matter? Would we see this in another type of malignancy or another situation? And we're now looking into that. And that's a good question, and that we really want to address that in the future.
Great, thank you. Antonio Casada is asking, he's from Switzerland, and he is asking, is there an endogenous anti-tumor T cell response primed after the transfer of serum antibodies from survivor mice?
Yes, we have not looked at that, but I suspect there probably is epitope spreading, and there might be an induction of host T cell responses.
Mm-hmm.
Yeah.
Sounds great. Xiao Dong is asking, how about the role of NK cells?
Yeah. So we have depleted the NK cells, and particularly in that lung met model, and just depleting the NK cells, and even in animals that don't get the serum, we get robust lung metastasis. Mm-hmm. We're trying to figure out a better way to address this question. I think just global depletion of NK cells maybe isn't the best way. I'd love their insight. I do think that they're important. And the reason why is by flow cytometry, we do see an increase in these NK cells. We've also found in the primary therapy, where we give anti-tumor Th17 cells and we deplete the NK cells, it's never significant, but there's a subtle decrease in the anti-tumor response. So I do think that there's some importance in NK cells, and we just need to do more work to really appreciate what that is.
Mhm. Sounds great. Great question, Xiaokun. Jaden Kim, I believe of Sinobiological, is asking, first of all, Jaden says, beautiful presentation. Let's see, it says, looking forward to hearing you speaking on this revolutionary work from the Palos Lab. And I think I'm just heading out to hear a presentation of yours in the near future. So it wasn't a question, but it was a nice compliment.
Oh, thank you, Jaden. You're awesome.
Thank you, Jaden. All right, and Omar Kassar has also got a follow-up question. CDR, B-cell receptor characterization and antigen determination. I think Omar's trying to clarify.
Oh, okay, sure. Yeah, so we're working on that now. We have a collaborator at Emory that specializes in that, and we just started a collaboration. We also have a collaboration that we just started with Aaron Ring. He has this really elegant work, which you guys might know about, where in different types of immunotherapies, there's an auto antibody response. And so it's not necessarily antigen specific, but there also might be other antibodies that are being produced that somehow contribute. So Aaron Ring has seen this in patients with checkpoint blockade. There's a certain signature of autoantibodies that are induced. So we're looking at that too. We just shipped him our serum all the way to Seattle, and he's looking at that now.
Okay, great. Glad to see Omar clarified his question there. We have a few more minutes if anybody would like to ask any follow-up questions. Let me just ask a very broad general one. I'm just curious about your goals for clinical trials, large scale, and what the timing is on that. And could you just tell us how close you are to kind of moving this towards the clinic?
Yeah, absolutely. So for the TIL project I was telling you about with Megan, we just got a grant from the V Foundation called the All-Star V Foundation grant. And so the goal of that grant is to really understand the cells better and to additional questions, but then also to then use that information to make an IND so we can treat patients with it. We're working on the IND now. We also have philanthropy to do so. So I project, hopefully if all goes well with all this paperwork and all, et cetera, that we'll be treating patients within a year's time.
Wow.
With the TIL product for patients with melanoma. We also have an R01 with Greg Lazinski as a contact PI to use these CAR T cells in patients with pancreatic cancer. And that's in partnership with the University of Pennsylvania. And I think we're around the same timeline of treating. Maybe that will be the first product and then the TIL will be the second product. It's not too far away.
Year time frame. That's very exciting.
Yeah,
sounds great. Well, I think we're wrapped up with the questions. We'll go ahead and finish up a few minutes early. I'll wait. One more question from Xiaokun, since we have the time. What cytokine or chemokine cocktail will be used to culture the TILs, TILS?
Yeah, so I have that in the publication. Let me see if I can go back though. So it's been published quite a long time now that IL-6 and TGF-beta are important for Th17 cells. So we use that. We also use IL-23 and IL-21, which are known to maintain them. Pavel Muransky had a paper in Immunity a long time ago, and we just borrowed his beautiful intellect into our situation and added IL-1 beta. It's complicated, right? We also inhibit interferon gamma. Initially, we inhibit IL-2 because it's been shown that it destroys our ability to become Th17 cells. So it's quite the cocktail. But this is all GMP cocktail that we are able to use for our TIL product. This is mouse work, of course, but it's not so different.
Very cool. We can make that stuff under GMP at Sino, just FYI.
Okay, great. I will note that.
One more question, we'll wrap it up. This looks like a good question from Jabezi Umuhire. Jabezi is asking, did you look at the balance or the imbalance between Th17 and Tregs in your mouse experiments?
Yes, we did. And we published it. It's in a cancer research paper by Hannah Knockelman is the first author. And there are Tregs. And in that particular paper, we found that one of the regulators of whether there were the amount of Tregs that were in this Th17 culture mattered. And so the majority of the adoptively transferred T cell product are Th17 cells, but there are some Tregs. So it's a good question. When we adoptively transfer these into mice, we see that I would say 95% of them have a Th17 phenotype, but about 5 to 10% of them look like they're regulatory T cells. Whether they're suppressive or not, we're not entirely sure, but that's driven by IL-6. And we published that work in, again, Cancer Research. I don't know if I reference it somewhere.
We had folks specifically, first and foremost, from Atlanta. I wonder why, Dr. Paulos. And we had folks from Sweden, as well as Poland, London, Korea, Budapest, Hungary, Cape Town, South Africa, and Macedonia. So folks really from all over. Also want to thank you, Dr. Paulos. Fantastic work. You truly, I think, are revolutionizing cancer therapy through your T cell studies. So congrats to you and your team and all your collaborators. It's really fascinating, and good luck to you in a year or so when you really start impacting patients. It's going to be fantastic.
Oh, thank you. Thank you for your time. And I thank everyone for attending. I know everyone's busy, so.
Yeah, it was a great turnout. And finally, thank my good friend and colleague, Kayla Sarmiento, who actually executed this webinar once again flawlessly. So thank you again, Kayla. And
yeah, thank you, Kayla.
With that, we'll sign off and say good afternoon or good evening to everybody.